We are recording.
We're good to go? All right. Sync test. Cool. That always feels silly. All right. Hey,
hello everyone.
welcome back, Bio 105 lab. We're doing the protein extraction and quantitation lab today. And there's a little bit of background information that we need to provide to make the lab make any sense at all. Mm-hmm. So our protein source is E. coli, correct?
So we have a little overnight culture of E. coli.
Yeah, we're going to be pulling protein out of it and seeing how much protein there is in E. coli cells, or how much we can get out of E. coli cells more accurately. So the first thing we have to talk about really briefly is a discussion of osmosis and tonicity. We may have talked about this in lecture at this point. Sometimes we don't get to it until a little bit later. Yeah. But what we've got here is a very, very interesting phenomenon, okay? When you have particles of solute dissolved in a solution, right? So, for example, salt in water, you actually absorb energy from the solution due to impact. It's a little complicated, but stick with me because it's better if you actually understand the real principle here.
He really likes this stuff. You can tell.
So here we go. What I have here is a membrane that water can move freely across, right? On both sides in this chamber, I have water molecules. And all of these molecules are moving around to some degree, okay? All the molecules are moving in completely random directions on both sides. And what ends up happening because of this, some molecules move this way, some molecules move this way, all right? But the net movement is the same. So the amount of water on each side of the membrane stays constant, right? But if you put in some molecules of solute, like sodium chloride, table salt, right? Then the water molecules actually encounter those, and as they deflect off them, they have less energy. It's a very interesting phenomenon. But what this means is that there is less movement of water molecules going on this side of the barrier, and more net movement on this side of the barrier. So, that random movement across the membrane is no longer at equilibrium, and we get more water flowing across in this direction. A little bit still moves that way, but not as much. So what this means, in osmosis, water will move from areas of low solute concentration to areas of high solute concentration. All that background that I laid out is not necessary for you to, pass a course or be able to describe the phenomenon, but it's always nice to know what's going on, energetically, right? So, all of this as background. Let's take a look at what happens to animal and plant cells when we put them into different types of solutions. Okay, so an animal cell, right? Clearly animal cells aren't just full of pure water, otherwise we'd just be a bunch of little sacks of water and we wouldn't be able to carry out tissue functions and all these important things. So there are particles of solute inside of an animal cell. If you put an animal cell into a solution of pure water, right? Water will be moving more into the cell. This is true of any solution that has a lower concentration of solute than inside of the cell, right? Some water technically does move out by random movement, but the net movement is into the cell, right? Now, the opposite is true if you put the cell into a solution with a lot of solute, right? We've already talked about it, so you should be able to predict this. Net water movement is now outside, or moving out of the cell, right? Mm-hmm. Fantastic. Now, this will continue until the concentrations of solute are equalized inside and outside of the cell. And this can occur if enough water is lost, because you get more concentrated inside the cell, you get less concentrated outside the cell. That's much more of a high-level concept, but you'll be fine with it, because I know you're doing a great job out there. All right, let's quickly talk about the language we use. All right.
Are you going to do the plant?
Yes, for sure. Yeah, we'll talk about the plant, the difference here, right here. we need to talk about the terminology of hyper, hypo, and isotonic. Hyper, like a hyperactive child, is more active. A hypertonic solution has more solute dissolved in it. Hypo, like a hypodermic needle that goes under your dermis, right? That has a lower concentration of solutes than whatever else we're discussing. Iso, like isometric exercises, the same, okay? This is how people try to sell something like coconut water. They call it isotonic, and it's supposed to hydrate you better for that purpose. Who knows if it works? So here's what happens. When we put these two different types of cells, the animal and the plant cell, into different types of solutions, right? Let's begin with a hypertonic solution. Why not, right? Hypertonic, high amount of solute outside of the cell. Water is lost, and the cell will shrivel up. Just shrivel up. Lose a lot of fluid, becomes like a weird, ugly little mass, right? Now, in a plant cell, this can still happen, but the cell membrane is held within a cell wall. So, you actually have the cell membrane begin to be drawn in from the cell wall. And we call this state plasmolysis, or the process plasmolysis. We call the cell plasmolysed. So, both things lose water in a hypertonic solution, but the cell wall leads to some differences. In a hypotonic solution, water wants to move into the cell. So, in an animal cell, water keeps moving in, pressure keeps building, it's like a water balloon. So then it becomes really massive, and then the cell will rip or lyse or split, and it will spill out its cell contents. In a plant cell, water will still move in, and it creates a lot of pressure, just like the water balloon over here, but this water balloon has box around it. So it won't actually explode. It actually generates something very important in plant structure called turgor pressure. So turgor pressure is pressure generated by fluids in this sort of process, and this is why a plant will wilt when you don't water it enough. All the cells are turgid, the plant stands up and it's happy, you start to lose water, and all of these cell membranes, they start to dry in, and you don't have the structure anymore provided by that turgor pressure. Plant wilts. Give it more water, cells draw up water, and then it becomes turgid and happy once again. Alright? I believe that runs through the background information that's necessary. Oh, in an isotonic solution, net movement's equal. Yeah, no big change there. So that covers that. We also do briefly need to discuss the importance of a detergent.
Yeah, so what would happen to a detergent in each of those cases?
Well, the detergent, it actually is useful in breaking up those regions of hydrophobicity, right? It breaks up the molecules comprising the membrane, the phospholipid bilayer.
Yeah.
So you get the same sort of process going, where the membrane dissolves away and cell contents are lost. We're trying to get at these proteins, see how many proteins we can extract, and we'll be using different solutions that have different tonicity values and that may or may not contain a detergent to see how well we can get all that good stuff out of some E. coli cells. We're not working with plant cells today, but but there will be labs where you see plasmolysed plant cells.
Okay.
Great.
So we're going to measure the protein concentration using an assay called the biosynchroninic assay. We're going to
really rolls off the tongue.
You do not have to remember that word. We're going to use the BCA assay to measure protein concentration. And that is a colorimetric assay where the solution contains copper in it. So it starts off slightly greenish, and in the presence of protein, you're going to produce a purple color. And the more purple it is, the more protein is present in that solution. So we call it a colorimetric assay. We'll use the Spectramax spectrophotometer again to do those readings. Right, and the purple color, the absorbance that we're going to measure is at 562 nanometers, is the wavelength of the light, the purple light that will get absorbed. Right?
Yeah, so this is only slightly different from the photosynthesis lab, which will occur in Bio 105. In that lab, we're measuring absorbance of the molecules directly. In this lab, there's a reaction that yields something else that we then measure. But it still represents the amount of protein in the sample.
All right. So, I think like we've covered the background.
I think we're good to go.
Okay.
All right.
So we have our sample of E. coli here, and we have 4 different solutions that you can't tell the difference between them. They're just labeled 1, 2, 3, and 4. But buffer number 1 is going to be a hypotonic.
So lower concentration of solute than within the E. coli cells.
Buffer number 2 is isotonic.
Same.
Buffer number 3, hypertonic.
Higher.
Okay. And buffer number 4 is isotonic with detergent.
All right.
All right. So we're going to compare which of those solutions gives the best yield of protein extraction in E. coli.
Perfect.
Okay, so we've got 4 tubes here. These are, I'm not sure that we've worked with these before, but these take a maximum of 1.5 milliliters, and
that's milliliters, so 1,500 microliters,
Yeah.
just to keep those units in mind.
And they're called anything in your protocols from flip-top tubes to
Centrifuge
ethanol
tubes.
to microtubes.
Yeah, they got all sorts of weird names.
Yeah, it just depends who wrote the protocol. Alright, so what we're going to do first is do the serial dilutions of BSA.
Perfect.
Alright,
So
so
what
we need 1, 2,
volumes do we need here?
7, 8. We are going to put 100 microliters of buffer number 2, which is the isotonic buffer, into 8 tubes.
Okay.
Okay, so what we're going to do at this point is generate some standards. Okay, so you guys have all had experience making a standard curve in the carbohydrate lab. We're going to do something similar here where we use a protein that we know the concentration of to create a standard curve, and then we will be able to compare the value in our E. coli sample and determine what the concentration of protein is in that.
Yeah, so we're just going to generate something like this. As the concentration of protein goes up, we expect absorbance values to increase. It's not always going to be a nice linear line like that, but that happens sometimes. And then, if we have any other sample, we can just measure absorbance values and find the intersect, and that will tell us protein concentrations. Just a quick refresher on the rationale for using a standard curve.
Yeah, so the protein that we have is BSA, which is bovine serum albumin. I'm going to write that out here.
So just to reiterate, we know the concentration of protein in this sample, which is why we can use it to generate a standard curve, which then can be used to measure unknown samples.
So the concentration in this little vial here is 2 milligrams per milliliter. So that will be our highest point over here, which is 2 milligram per milliliter. And what we're going to do is we're going to do a dilution series, where each time we dilute this by half, right?
Yeah.
So our points are going to be, starting at 2, we're going to have 1, 0, 5, and this is not the correct scale.
No. Don't hold us to this drawing.
0.25, 0.125, 0.6 something, and 0.3 something.
Yeah. So we call this a serial reducing solution. Is that the terminology people use sometimes?
Yeah.
Yeah. And it's easy enough to see how this works. You're starting with 2 milligrams per milliliter, right? And you're doing a 1/2 dilution. Oops, excuse me. So the math there works out to tell you that it's a 1 milligram per every milliliter solution now. And we can using the volume that we add in to each of these tubes, we will know exactly how much protein's in that entire container. Yeah.
All right, so if you look in your protocol on page 3, there is a table, and it starts with the stock solution of BSA, and you're going to take 100 microliters out of that stock and add it to 100 microliters of buffer, which is essentially a 1-to-1 dilution.
No.
Okay. All right.
And,
so
and also that would work out for the 1/2 dilution because it's your.
Sorry.
No problem. No problem. We're dancing around. It's fine. We got 1 milliliter of our solution, starting solution, over the total volume of the solution, which is 1 milliliter starting solution plus 1 milliliter of our diluent, or what we're diluting with.
Yeah.
Just in case you're a fan of these dilution factor things, might as well throw it up there.
All right. So do you have the pipette?
I sure do, and it's set to 200 for the first tube, correct?
Actually, no. I think so. I'm going to start off by just adding 100 microliters into each of the tubes.
Okay, yeah.
So I'm using the P200, which can go up to a total of 200 microliters. We only need it set at 100 right now. So there it is, set at 100 microliters. And what I'm going to do is I'm going to take buffer number 2, which is the isotonic one.
And so it doesn't go into tube A though. We have 0 microliters,
Yeah.
so every other tube though gets 100.
Okay.
This is what bench work usually looks like. Too many cooks in the kitchen.
All right, so I'm just adding the 100 to each of them. Got a bubble in there.
Oh.
I think I need one more tube.
Oh, you need an extra
Yeah.
flip top. Get you one of those. Here you are.
All right. Now I'm going to put 200 microliters into tube number A. Okay. So this is the BSA, 2 mg per ml stock.
Sometimes you just have an urge to smell things in this lab, and I don't know why I'm curious what aroma that would have.
I don't think it has any smell
Nothing?
at all, but
Oh.
go ahead.
That's bad, yeah. With the lab technique.
Alright, so we've got our stock. I'm going to label it. I don't want to get confused here. It's really easy.
Oh yeah, I'm happy to label tubes while you create them if
Okay.
you like.
Alright, so now what I'm going to do is out of that first tube, I'm going to take 100 microliters and add it to the 100 microliters of buffer number 2, which will be diluting it.
Here we go.
Okay, so set on 100 microliters now. Take 100 out.
So we're going from 2 milligrams protein per milliliter, diluting it by 1/2. So this now will have.
See that there's 100 in there now.
Brand
Add
now it's up
another
to
100 to it.
Yeah.
And now we're up to 2.
Stupendous.
Alright, now I'm going to vortex to mix.
Mix it up.
Okay. Now I'm going to take 100 out of there and put it in my next tube. Okay.
Yep. So now this had 1 milligram per milliliter, and now we're diluting it with another 1/2 dilution. So now we're down to 0.5.
Okay. And vortex again. Right.
Looks good to
We'll
me.
do it again. Usually you would probably want to change tips. I'm economizing here. 'Cause
Yeah.
It's just for demonstration. So we're saving our planet.
Yes,
One
exactly.
tip at a time. Okay, there's C.
Stupendous. Yeah, we will always clarify if we're doing something slightly different from how you would do a normal lab, due to the fact that these are demonstration labs. But we'll try not to, try not to rob you of any precious information.
There you go.
Cool, thank you.
Alright. C.
So Bridget and I were talking about this. This is kind of like one of those infomercial, shows that used to be on at 1 in the morning or something, where they're trying to sell you a fancy juicer.
Yeah, really low peak hours.
Yeah. Hey, have you ever purchased anything off of one of those?
No, I haven't.
Me neither. Somebody has.
Yeah, they must, right?
They have to, yeah. I don't even know if those are still on the television. I think it's all streaming now. And Instagram ads, I guess. I don't understand the world these days.
Somewhere between that and a cooking show, maybe.
Yeah, exactly.
Alright, so we have made a dilution series
Look at that.
of doubling dilutions.
They are labeled. We have a corresponding table telling us what the concentrations are now. So, there she blows, yeah.
And as an exercise, when you are looking at this, I would recommend going through and in your table writing those concentrations.
Yeah.
of BCA for
You
each.
definitely really want to get comfortable with the dilution math. That's a thing that a lot of people stumble on in this course. So doing some practice, talking to us as always, talk to your TAs, do the dilution practice problems that are in your lab main protocol, all that stuff. Heck, just go online and look for dilution practice problems. It gets to be fun after a while.
Alright, so now we turn the page here,
Yep.
and we are going to retrieve our E. coli sample.
Perfect. That would be this one,
Yeah.
correct?
So we have it sitting on ice here, just to keep it nice and happy while we are
Yes.
messing around. Okay, and we need 4 tubes.
Okay, cool. So we're going to be using, while we're attempting to lyse these cells, first we want to get all the cells concentrated so they're not all spread out through a solution, right? So a classic way of doing that, of concentrating things out of a solution, or out of a mixture to be accurate, is with centrifugation. So that's what we'll be doing right here.
Alright, so here's the culture of E. coli. I'm using the P200 again, set on 200 microliters.
Perfect.
And I'm going to aliquot 200 microliters into each of the 4 tubes. 1, 2, 3, and 4. So we're just going to number them 1, 2, 3, and 4, which will correspond with the 4 extraction buffers.
Yeah. So
Okay,
always
this has E. coli on it, so I'm going to put it in the biohazard bin.
So all of that work we did before, remember, that was all with bovine serum albumin. So this step here is where we're actually taking E. coli cells and we're going to split them open using different buffers and see how efficient we are at retrieving proteins. It's always good to double check why we're doing what we're doing as you run through a protocol like this. It's easy enough to get blinders on and just start going.
Alright, so we're going to pop them in the centrifuge.
Yep, we'll drop them right in here. It's always important to balance your centrifuge. Otherwise, you can get some wobble on it. I will take a quick picture, actually.
Awesome.
Want to make sure that we have all the information for you so you can see what this looks like. There we go.
Alright, and so what do we need to spin it at? Maximum speed for 3 minutes. 3.
Oh yeah, these ones have rough lids. Nice. Alright. So, I'll probably edit this part out of the video, but if I don't, then
We'll just be standing here.
we'll just be hanging out waiting for the centrifuge.
So what do we do?
Yeah, let's just look ahead. Okay, yeah, so we
So we're going to add 200 microliters of the vapor.
mix it up,
Or next.
mix it on ice, then pour it into the centrifuge. Okay,
And
okay.
then dilute it.
Perfect. Do we remove the fluid on in this way? I don't think we do.
Yeah, we do.
We do? Okay. There.
Should we get a container?
Oh wait, we use the liquid. We get rid of the debris at the bottom.
Yeah, but at this point we're supposed to take off the liquid too.
Oh,
We could
So, just to say it one more time, we've got our original sample here, which is our E. coli culture. Sorry for yelling at you, I want to make sure you can hear me over the centrifuge. Now the E. coli cells are kind of dispersed through there. So what we're doing here is getting all the cells concentrated into one little region. We're going to dump off the fluid that these are swimming in right now. We're going to put in all these buffers that we've been talking about, right? And then mix that up again with the pellet, and that will allow the buffer to interact in a concentrated way with the cells. I'll go ahead and put one right in front of the camera so we can see what a pellet looks like. Okay, how's this look? So you should be able to see right there, there's a clear fluid and then a little tiny pellet, looks like a little booger at the bottom. I know that doesn't look perfect, but bear with us, please. Okay, so that's all the E. coli cells, and we do not need the fluid at this time. So where Bridget right there is pulling the fluid off, being careful not to lose our pellet full of all our precious E. coli.
And I'm putting it in another tube just to store the waste. Okay.
Perfect.
So now you can add 200 microliters of each of the 4.
Okay, you got it. Oh, actually, I need another P or I can use this P1000. Okay, do.
Okay.
And are those
So
buffers
now
in order from 1 through
yeah.
4? Awesome.
Now we have just the pellet of cells with no fluid on it anymore.
Oh,
Sorry.
that's okay.
Just lodge that.
Yeah, pellet's fine. As long as the pellet's fine.
We're going to bring the biohazard bin over.
That's not a bad idea at all. Now, this is a non-virulent strain of E. coli, right?
Yeah.
So, it could be worse. Okay. Once again, economizing on tips. Normally, I would be changing the tip out between all of these different buffers, but this is for demonstration purposes. And then we get to vortex, don't we?
Yeah.
Tip disposal.
Here you go.
Oh, thank you.
Okay. And then vortex. Try and resuspend. We can do them all at the same time.
Yeah. They always give us a bit of a hard time.
We could use a pipette to resuspend these.
That's true.
The pellets are not very compact. They're not wanting to go into the
Yeah. Well, since it is a demonstration, we don't necessarily have to get all of them completely resuspended. But yeah, that's a classic technique actually to mix a solution. Oftentimes you'll just take a pipette, and I can demonstrate with one. Yeah. You'll take your pipette, and you'll just put it in there, and just draw it up and eject it just over and over again. And that's a very good way to resuspend a pellet. It's a great way to get everything really mixed up. It can actually be damaging, so you don't always want to do it with a sample. But with this, we're literally trying to break these cells open, so I think we'll be okay.
Yeah. All right, let's put them on ice for 5 minutes.
Stupendous.
Okay.
Oh, let's make sure it doesn't affect the filming.
Yeah, I think it's okay.
I'll actually just double check. Awesome. Everything's working. It's so good to see.
All right, while we're waiting for that 5 minutes, let us label another 4 tubes.
All right.
1D, 2D, 3D, and 4D.
1D, 2D, 3D, 4D.
So what we're going to do in those tubes is, because if our protein concentration is higher than 2 milligrams per mil in our extracted sample, it might be off the curve, right? And so we want to make sure that what we're measuring is within the range of 0.3 to 2 milligrams per mil, right? So we need to do one sample where it's concentrated, and then we'll do another one where it's diluted.
Yeah.
Okay.
And the goal is really just to get something here, and then because we know how diluted our samples were, we can sort of backtrack the calculation.
Yeah.
To any volume.
So the dilution we're going to do here is a 1 in 5 dilution, and so we're going to take 20 microliters of our protein. We're going to add it, so that's of our protein. Whoops.
It's always hard to write across this divider here.
And we're going to have 80 microliters of diluent, so solution. Alright, so that gives you a total of 100 microliters. So 20 and 100 is a 1 in 5 dilution.
Yeah. So it's whatever you're diluting over the total volume. So the volume that you create from what's being diluted and what is doing the diluting. And that gives you your dilution factor. So there it is in two very clear explanations, I think.
Okay,
Awesome.
so let's put the 80 microliters of each of the buffers. So
Brilliant.
we need 80 microliters of buffer 1, buffer 2, buffer 3, and buffer 4 in each of the 4 tubes.
Perfect. They're opened and in order.
I'm going to change this to 80 microliters. And
Once again, these tips would be changed out in a normal lab.
yeah, definitely.
Which is why the tip boxes are so handy dandy. You can really rapidly swap tips out once you get a grip on it. Cool.
Okay, how
That's
we
our
doing?
80.
We've got 2 minutes left.
Perfect. I would say that we're fine to just keep moving through it, yeah.
Okay.
Okay, so now we just need to centrifuge at full speed for 5 minutes.
Okay.
Cool. So what's happened here now is the cells have been put into the solution with the buffers. So the buffers are all acting as described earlier. The detergents are breaking down membranes. The hypertonic solution should be bursting, the hypotonic solution should be bursting these cells. And now we want to actually take all the cellular debris from the broken open cells, and we want to pull that out. So that's what's going to happen here. Let's see how much of that sound's getting picked up. Yeah. It's not popping the microphone or anything, but they might have a hard time hearing that sort of centrifuge. If you can't tell, this is our first time sort of doing a lab this way. I need to plug the laptop in.
You're going to edit this out, right?
Yeah. Or, there's always a chance I'll forget it, but I think it's humanizing, right?